Hello, everyone. My name is Emile Sylvestre. I'm an assistant professor in civil engineering at the Delft University of Technology and formerly a postdoctoral fellow at the EVA. The title of my presentation today is Quantification of Legionella pneumophila in Building Potable Water Systems, a meta-analysis comparing qPCR and culture-based detection methods. In this video, I'll present you my work, which is part of work package 3 of the LeCo project, which focus on detection methods. To understand the challenges in detecting Legionella pneumophila and why method selection matters for risk assessment, let's first look at the two main quantification methods used in environmental monitoring. Culture-based methods, such as ISO 11731, quantify viable bacteria in colony-forming units, and these results can be directly linked to infection risk estimates through available dose response models. However, cultivation is labor-intensive and slow, requiring 8 to 14 days of incubation. In contrast, standard qPCR methods provide faster results and can even be automated for on-site implementation. But they can quantify genome copies without distinguishing live from dead cells, potentially overestimating health risk. To help interpret qPCR data in Legionella risk assessment, the key question we asked was, what is the relationship between results obtained with cultivation and qPCR methods? Let's now look at the factors that influence that relationship. First, differences between analytical methods are expected to lead to different results, as they can have different measurement error and recovery efficiencies. Equally important, environmental factors such as heat and chemical treatment can significantly impact the relationship because they affect bacterial viability, but do not necessarily degrade their DNA. So to accurately quantify the qPCR to culture relationship, we must account for both methodological and environmental drivers. The aim of this work was to compare qPCR and culture-based detection methods for Legionella pneumophila in potable water system. More specifically, our three objectives were first, model how the qPCR to culture concentration ratio behaves within individual buildings over time. Second, to meta-analyze these building-specific ratios to see what pattern emerge across different systems. And third, deliver conversion factor that help integrate qPCR data into risk assessment, especially when culture-based results are not available. To build our database, we conducted a systematic review of studies comparing qPCR and culture results from Legionella pneumophila in potable water systems. We screened over 1,500 records from Web of Science, PubMed, and Scopus, along with references from existing reviews. After removing duplicates, about 1,100 records were screened based on title and abstract. Only 189 full-text articles were assessed in detail for eligibility, and in the end, 15 studies were included in the final analysis. Of these, 5 studies provide building-specific data sets, and 10 reported pool results across multiple sites. The final selection formed the basis for the meta-analysis I will show you next. From the selected studies, we extracted a set of key parameters needed to characterize the water systems, treatment condition, and measurement methods. These include information on the water system type, preventive or curative control measures, and water quality indicators such as chlorine level and temperature. We also collected details about sampling processing and measurement performance, like sample volume, recovery rates, and detection limits. However, some information, especially about disinfection treatments, was only partly available, being reported in roughly 20% of the papers. This gap introduced some uncertainty when we tried to link environmental condition to qPCR to culture relationship. To capture how Legionella pneumophila concentration vary over time and space within a building, we use Poisson log normal models for both cultivation and qPCR data. This model is very useful as it can handle Legionella counts that are below detection limits, as well as the high variability in Legionella concentration that is often observed in water system. This modeling approach allowed us to model the relationship between qPCR and culture results as the ratio of two log normal variables. The detailed formula on the slide show the mathematical structure, but the important point is that this approach allowed us to predict how variable is the qPCR to culture ratio and what is the uncertainty about this variability. So here's an example of application of the model to data from a Swiss hospital. Panel A showed the usual log-log regression between qPCR and culture results with a correlation coefficient of 0.45. We can see that while this regression line suggests some relationship, it does not provide much information about how this relationship vary over time. Panel B showed a cumulative distribution of the concentration measured by each method separately. We can see that qPCR generally detect higher concentration than culture, and that qPCR data seems to be more variable than culture data. The key insights come from panel C, where we model the distribution of the qPCR to culture ratio. Here, we can clearly see that at this single site, the ratio between qPCR and culture varies by over 3 orders of magnitudes, so from below 1 to well above 1,000. This highlights that the relationship between qPCR and culture result is not fixed, but can fluctuate significantly within the same building over time. Accounting for this variability is essential for developing safe and realistic conversion factor to input qPCR data into risk assessment. To compare results across different studies and sites, we extracted two types of average ratios predicted by this model. First, the geometric mean ratio, which reflects the typical value expected at the site. And second, the arithmetic mean ratio, which is much more sensitive to high ratios, like occasional peaks of DNA gene copies without culturable bacteria. We also calculate the standard errors for both types of mean, which allowed us to properly weight each study in the meta-analysis. The important advantage of this method is that we can compare ratio across diverse buildings and system condition. Here we see the main results of the meta-analysis, showing the geometric mean in green and the arithmetic mean ratio in blue for each study site. The dotted vertical line represents a 1-to-1 ratio, where qPCR and culture concentration are on average equal. What we can see is that several studies cluster near the 1-to-1 line, especially when looking at the geometric means. About 80% of the sites show a geometric mean ratio below 10-to-1, meaning that typically qPCR measurements are within 1 order of magnitude of culture measurements. However, there's also a few studies, particularly when we're considering the arithmetic mean, that show much higher ratio. These high ratio are driven by occasional very high ratio where gene copy are much higher than viable bacteria. The key takeaway is that while there's variability, for most sites, a simple default factor close to 1-to-1 is reasonable for interpreting qPCR data and risk assessment. Nevertheless, some studies deviate more strongly from the 1-to-1 line, and the application of the 1-to-1 factor could lead to highly conservative risk estimates. Now let's have a look at one potential moderator of this relationship. The slides show an example from 3 non-chlorinated hospitals, focusing on how system condition affects the qPCR to culture ratios. In panel A, we see the cumulative distribution of the qPCR to culture ratio for the 3 buildings. We can see that the distribution of the ratio varies substantially between sites. Panel B shows the distribution of water temperature recorded at each site. We can see that the system with the narrowest and highest temperature, system 100, also has the most stable and lowest qPCR to culture ratios, clustered close to 1:1. In contrast, the system with wider and lower temperature distribution, system 110 and system 120, show much higher and more variable ratios. This illustrates that the control of water temperature is a major factor affecting the stability of the qPCR culture relationship. It should be noted that other factors likely influence ratios. For example, shock disinfection events can cause temporary spike in ratios. However, based on the available data, in-depth analysis of such events was not possible. Now, to conclude, first, qPCR to culture ratio can vary both within a single building and between different systems. Based on our meta-analysis, we recommend applying a conservative 1:1 ratio between qPCR and culture results when quantitative risk assessment must rely on qPCR measurement alone. We found that tight temperature control stabilized the relationship between qPCR and culture-based measurements, supporting the importance of good operational management. Other influences, such as shock disinfection events, can also affect this relationship, but further standardized research is needed before specific adjustment can be recommended. Finally, it's important to keep in mind that high qPCR results suggest past or ongoing Legionella growth in the system. Thus, interpreting these results for risk management decision required not only considering the number themselves, but also understanding the operational history and system condition behind them. This is the end of my presentation. Thank you for your attention. If you found this presentation interesting and have specific question, feel free to contact me directly or visit the main page and post your question using the question and answers link. Also, if you would like to learn more about the LeCo project, be sure to check out the other videos.